So, as the title say, with Mark Whiteman, we invented the ultramicroelectrode to study cells and brain tissues. And let's start with that. Everybody of us have seen this kind of picture into the literature. This is the brain of man listening to the music, like me, okay? So this is the left map of the NMR brain map. And now he studies as analytical, and you see that the centers in the brain are different. Because the analytical, this guy is a professional violinist. Now he's thinking about the music in order to reproduce it. And in fact, that it is, we see these pictures, but it's never explained what we are seeing truly. What we are seeing is that it is a concretization of a former and old, this is not a mistake, it is from 1890, okay, the paper. From Roy and Sherrington, who understood that the brain is vascularized, and since it's vascularized, when the neurons are working, they ask for glucose and oxygen and so on in order to perform their function. So, in fact, the brain will then increase the flow of blood where it works and decrease where it does not work, because the case of the skull is fixed, so you cannot make overpressure or under pressure. So, in fact, basically, this is that. You see if on the left, the neurons are working, okay, they exchange the classical neurotransmitter and so on. They sent hypothesis from Roy and Sherrington. They sent nitric oxide, and in return, the nitric oxide will provoke a dilatation of the epithelial cells of the capillaries, and the lumen will enlarge, so there will be more blood locally. So that has remained in, as an hypothesis up to the point where we could study that. And to study that, we introduced the very small electrode. So for this, now we do nanometric electrode, you will see in the second talk. But for that, we needed to have electrodes that are about 1 to 5 micrometer at the tip. So it's carbon fiber, which is etched, insulated with a polymer, and then cut at the tip, so we can have the size we want. And then we platinize, so the carbon is not active with the reactive oxygen species and reactive nitrogen species. So we have to deposit platinum, black platinum. So here you have on the photograph, you have an electrode with the deposited platinum, okay, at the tip. And otherwise, what we can do is have a tube, a micropipette, kind of a Pasteur micropipette, and inside we deposit the platinum. And here you have a scan that shows that we can really deposit the platinum over nanometric scales. So with those, you can bring the electrode in the same way as in a synapse. So the cell is emitting things. The microelectrode receives the thing in the same way as in a synapse. When you have the emitting neuron, sends the neurotransmitter, and the receptor cell receives. Okay, so it's exactly that. But now we can do that with macrophage, for example, or different cells, or in the brain, and we stimulate, and there are a very small flux of reactive oxygen species and reactive nitrogen species. This is a curve like this, a burst like this, shown on the right. This is about 3,000 molecules. When I say 3,000 molecules, this is really 3,000 molecules. So, since the different reactive oxygen species and nitrogen species are active at different potential. By changing the potential of the electrode, we can monitor the emission by the ear of the murine macrophage of the nitric oxide and the superoxide. And this is very small. You see 30 femtomoles per cell, okay? This is extremely small quantities, but for a cell is very big. So as you have seen from the talk this morning, when you have, when you have creation of superoxide by NADPH oxidase and O2, and O2 with NO synthase and L-arginine, that was in the talk of Farid Mourad, okay, gives nitric oxide. And now superoxide can disproportionate to give you hydrogen peroxide, so this is what everybody says, okay? And then Fenton reaction give OH dot, okay? Or NO can go out, this is the bottom one here. But when both the superoxide and the nitric oxide meet, they generate peroxynitrite. And peroxynitrite, a part of it will die while going out of the cell, generating nitrate on NO2 minus ions, or go outside as peroxynitrite. And that will generate OH dot and radical, free radical in the lipidic environment. So I will not comment the homeostatic balance because we had several talks where this concept was used. But basically, if we don't have enough of oxidative stress, we cannot live because any bacteria and so on can attack us and we will be losing, okay? But if we have too much, this is us. So we need to have a balance that is perfectly centered, and this is what is called homeostasis. Now, if we want to see that in the brain, okay, the experiments are done this way. This is a rat brain, okay, cartoon, okay. You have the cerebellum, and we work in slice of the cerebellum. The slices are a bit thick compared to what biologists are using, because we want that the cell in the middle, when the slice is perfused, the cell in the middle believes that they are still in the cerebellum. So they don't realize that they are cut from the cerebellum, okay? So what you see is exactly that. On the, for example, you have on the middle, you have a micrograph of the brain slice, okay? On the left, you see in red is a color after the experiment. You have the capillaries that are here, okay? And you see, it's interesting to see that the capillaries are equidistant of about 50 micrometer in the brain of all the mammals, okay, including us. And in green, you have stellate neuron. These are the more important neuron in the cerebellum. So, with on the side, in the blue square, you have the microelectrodes that I have shown before, which is close to the system. And now we can do two things. With the electrode, we can monitor the chemical nature of the oxidative stress and the reactive nitrogen species. And with the optical microscope, we can look simply by changing the kind of light, okay? We can see the capillary here, the one in red where you have the arrows here. In the normal state is shrink, so the red cell cannot carry, okay? They are blocked, but you can keep the homeostasis for the fluids, so sodium, potassium, and all that, okay? Now, when you stimulate the brain locally, what you have is the kink is opening, the red cell can carry. Okay, these are part of a movie, but when in the movie you see the red cells moving, And now when you are cutting the stimulation of the neuron, the green one, is stimulated with a patch clamp, okay? So when you stop the stimulation, the neuron stops to work, and the vessel close again. So you are back to the normal, and the flux, this is what you see here. This is an enlargement. You have the electrode. You have the patch clamp that stimulates the neuron just in the kind of ellipse, which is here, is the body of the neuron. And you can detect with the electrode the rise of the nitric oxide. And when the stimulation ends, the nitric oxide goes down. And in parallel, you can see the opening of the kink here. You see that is opening above like something in 10%. This is enough for letting the red cells and glucose and so on to pass, oxygen and so on. What is interesting is that around the neuron, the concentration of the nitric oxide will decrease simply by diffusion. And if you look what you have, this is exactly the same slide as before. The red thing are the region where one neuron will hit either the capillary, which is going straight like this, on the left or on the right. So this means that when neurons in this region work, they can ask for food and oxygen in this region, okay? So now, what is the relationship with Alzheimer? Okay, this is just a cartoon to remember what it is, okay? This is very scary because when you have Alzheimer, you don't even realize that you have Alzheimer, okay? People around you realize, but this is scary, okay? So as you know, people have been focusing on the plaques. Plaques are, you have this amyloid precursor protein, which is on the left top, then is cut here and release this amyloid-β 42. 42 because you have 42 peptides. And in the cartoon, you see what happens. These amyloid-β are clustering together and make a plaque. So the doctor, the medical doctor, said the plaque is making the problem, okay? And in fact, here on the orange one, you have a section of such a thing where the big dark orange circle is the cross-section of a plaque. So people are saying, okay, the plaques are creating a problem, so that what they try to do is to dissolve the plaque. And by doing that, you worsen the system. Why? Because when you have one peptide, amyloid-β 42, some are containing copper 2, okay? And chemically, the copper 2 is placed in a system that is catch here. The one in the middle is a DFT calculation, and the one on the right is the cartoon made by chemical thing. And this copper can be easily reduced to copper 1. And when this is what you have on the bottom, the copper 2 plus ascorbic acid, okay? So that's the reason why ascorbate is an antioxidant, but is also a pro-oxidant. Because it will reduce the copper 1, and now copper 1, okay, will react with the oxygen, okay? Because copper 1 is very active. So you have to remember that if we go back to the working of the brain, we have a flux of oxygen coming out of the capillaries when you have a flux of nitric oxide sent by the neuron to speak with the capillaries. As a result, you have something like is on the cartoon on the right, where you have the amyloid-β with its copper now reduced by the ascorbate to copper 1, and now this copper 1 will react directly with oxygen. This is a classical reaction in chemistry. Copper 1 plus oxygen gives a copper, gives this one. Sorry, I will show. So this one here now is a radical, like Professor Yoshikawa has explained this morning, okay? This radical will react with the nitric oxide, which is also a radical, and they couple. And now, if you know chemistry, you recognize this is a copper 2 attached to peroxynitrite. So again, another reduction, and you release the peroxynitrite. This is the one in the red, okay, here. And that regenerates the copper 2, okay, which is reduced again, okay, and you rotate that. All the structure here have been computed by DFT, so it's not something written by hand, okay. We know all the energies and so on. So this cycle, you need only a very small amount of the amyloid-β, but the key thing, no, but it's okay. What it is important is that you need to do that, you need that the amyloid-β is able to adapt the conformation when it is reduced, and adapt also the conformation when you have the bond with oxygen. So when you have the plaque, they are all like the pages of a book, okay? So with that, you cannot make this reaction. So in fact, my model is that the plaque is a way for the brain to protect itself, okay, to eliminate. But the culprit is amyloid-β itself. So what now, you have this peroxynitrite which is released. Peroxynitrite is lipophilic and hydrophilic. So it can very quickly enter into the lipids of the membrane, and when it is in that, we have computed, still by DFT, that it breaks spontaneously with a reaction which is 0 in delta G, okay, and a barrier which is almost 0. It gives you one radical hydroxy and one NO2 dot, okay? And in fact, what you have is you have the peroxynitrite, which is here in energy. It gives OH, which is very high, but this is compensated by NO2 dot, which is low. So in fact, the energy remains all around 0, and you have that. So now, one OH dot here is right. Inside of the lipidic chain, it will grab one hydrogen, okay, to make H2O, okay? Now, this radical reacts with oxygen, makes a peroxyl radical that grabs to an hydrogen to the next one, okay? And here you have a cycle that starts like this. So you have only a few OH dots are sufficient to create a big hole, okay, in the membrane. When the membrane has this big hole created, okay, it cannot maintain its membrane potential, it cannot maintain its equilibrium with the external side. And in the axon, as you well know, the propagation of the nerve influx requires this fine control of the potassium-sodium content. So when you have that, the axon starts to die and the neuron dies, okay? So this is a model. Unfortunately, I don't know how we can prove that in the experiment, but today the calculation, the DFT calculation are sufficient that we can do that. So in fact, this will lead to apoptosis. This mechanism has to do like this. And the key species is the formation of this peroxynitrite. So when you have here, I think since the audience is used to work with nitric oxide and so on, okay? So on the top, you have what is happening when you have apoptosis. You see this is a cell membrane that is completely cut into pieces, and that is a human cell. In fact, I believe this is a fibroblast. that is completely cut into pieces, and then it will be eaten. So it suffers exactly what happens to bacteria when they are during the phagocytosis. And that explains perfectly what you find in the brain of patient. In the healthy brain, okay, we have cavities, but in the advanced Alzheimer, these cavities are completely enlarged because the matter has been replaced by water, okay? In fact, you see exactly what is happening. The white and gray matter has been eaten and is replaced by extracellular fluid. So this is a model, okay? I cannot say it is a proof, but this is, in my view, the only model that explains everything we know about Alzheimer. So if we want to do something, we need to do something that will be coordinating the copper to avoid that it is reduced, okay? To conclude, I want to thank all the people who worked with me. So this started in my lab with Dr. Manongi, that was her PhD. And you have Professor Irina Svir, who is here, Dr. Alexander Olinik, who is my chairman. For the microelectrode, we have two kinds of collaboration. One for the electrodes that are micropipettes filled with the CVD carbon and platinum. This with the team of Mikhail Mirkin in New York. And the experiments were done essentially by Jixian Wang, who was doing a PhD at this moment. The experiment in the brain slice were done by collaboration with my friend Professor Jean Rossier, who is also academician of France like me, and the student at the time was Armel Rassiliac. All the calculations were done by Roberto Giacovazzi in the lab of my friend Carlo Adamo, who is one of the people who contributed to make Gaussian. So we are sure that the calculations are correct. And in China, this is a microelectrode that we use for checking inside of the cell. This is with Professor Weiwa Wang, who will give a talk on Wednesday online. And the experiments were done by Qinwei Zhang and assisted by Professor Yang-Ling Yu. So finally, you have on the bottom of the slide all the people who, the institution who finance this work. So École Normale Supérieure, the Xiamen University, because now I am also professor in Xiamen University. In France, I am emeritus professor. CNRS, Wuhan University, the joint lab of France with Wuhan and Xiamen and École Normale Supérieure, the Ministry of Research, French, the PSL University, Sorbonne University, and the NSFC who gives the money in China. And on the side here, you have all the academies in which I belonged up to now. It was a great pleasure for me to give this talk in front of you. And if you have comments, you are welcome. Thank you very much. Now you
thank you very much, Christian, for this very interesting talk. And if I understand correctly, we don't have comments now, so I will. Anyway, you can discuss during the coffee break, etc. And it's again my pleasure to present the second talk by Professor Christian Amatore. And this time the talk concerns about measurement of the ROS and RNS at the cellular and subcellular level. So please, Christian.